Good morning, everyone. Welcome to the latest webinar by MBF Bioscience. I'm Sue Tappan, staff scientist here at MBF. I also run the day-to-day operations at our contract research facility, MBF Labs. I'm joined today by Dr. Mark West, a renowned stereologist from Aarhus University in Denmark. This is our first multi-continental webinar. Mark, what time is it there in Aarhus?
Yeah, well, now it's just a little bit after 5 in the afternoon, so we're a little bit behind you. But we're ready, and thank you for inviting me here. This is really a great opportunity to talk about things that I think are actually very important. So I'm looking forward to getting into this issue of what is a bias and in stereology and what you can actually do about it.
Great. So let's get right to it. Dr. West is a professor of neurobiology at Aarhus University in Denmark. And you started teaching stereology how long ago?
Yeah, way back in the late 80s when there was a group here in Aarhus that was involved in the development of these so-called unbiased stereological techniques. And I was fortunate enough to be associated with them. So actually, so that goes back, yeah, what is that? That would be 12 and 14, 26 years. I've been teaching this type of stereology, but actually I started doing old-fashioned stereology when I was a graduate student, and then realized how new and important that sort of stereology that we're going to be talking about this afternoon is in terms of its value to scientific experimentation.
Yeah, so you've published many papers on the topic, and importantly, you've been involved in the development of some of the most commonly used stereologic probes in biological sciences. You've had a hand in the development of the optical dissector, the optical fractionator, and the Spaceballs probe.
Well, that's correct. I was fortunate to be associated with a lot of other people who had good ideas about this. My own particular interest in it was actually using these methods. I'm not initially a sort of theoretical stereologist, but I realized how important these things were, so I sort of came in the back door and started applying them. People started paying attention to this particular approach that we're going to be talking about today.
And that has led you to write a book?
Yes, this relatively recently I have been teaching the stereology at different places, amongst them the Marine Biology Laboratory in Woods Hole, and often in connection with the neuroscience meetings. And over the years, sort of a sequence of presentations that hung together, that presented a lot of these new concepts and practical applications of them. I decided to put it down once and for all in one place. So that was sort of what you see here, basic stereology for biologists and neuroscientists. Most of the examples are from the brain, but they're really applicable to any histological organ.
I think it's fantastic. And I'm really pleased that you're joining me today. You're taking time out of your busy schedule to work with me on this topic, which I think we are both very passionate and very excited about. And I want to thank the attendees for joining us also. I want to take a few moments to cover some of the basic information about how the webinar is going to proceed, so that we can get information from the participants as they have questions and so on. So in general, this webinar is being recorded and will be available to view later. A link to the webinar will be sent to you in an email. If you'd like a certificate of completion for attending this webinar, please let us know in the question window and we'll provide one to you. All questions and answers will be available on our website after the webinar, even those that aren't answered live. And a key to a successful webinar today is going to be your questions. So let me take a minute to go over how to send them to us. To submit questions to the presenter, you're going to simply click that orange arrow button to open the GoToWebinar control panel. Find the question tool panel, type a question in the box, and click send. And just undock that question window if you want more space from that panel if desired. as you submit questions, if you wouldn't mind if you could put all of the question text in a single entry, that will make it easier for us to read the entirety of your thoughts before other questions come in and separate them in the queue. And so if you could do that before you hit send, that would be greatly appreciated. So today we're going to be talking about biases in stereology. And since stereology primarily involves statistical estimates of total quantities of structural parameters, we're going to be talking about statistical biases. Our conversation today will touch on many things that can cause bias when it comes to designing your experiment. We're going to spend the majority of the discussion focusing on how to use sampling techniques to avoid biases so that the data you obtain is reliable, which makes the conclusions you make from your data more sound. So first we're going to cover the concepts of variability, precision, and bias. Then we're going to dive into those sources of bias and how proper implementation of stereology results in unbiased data. We're going to pause periodically to answer questions that you're going to submit throughout this webinar. So let's get right to it. So Mark, would you like to start us off?
Yeah, well, let me just first say in the beginning here, this illustrates what a bias is. And you can see here we have a cluster of samples that are made that are on mark, that is to say they're around the truth, and on average would be the truth. And here we have something that systematically deviates, and regardless of how many samples we make, we're going to have samples that don't really represent the true value of what we're trying to estimate. So it's a systematic deviation from a true value. But one of the things we're going to try to make sure people understand is that you also have variability in the estimate itself. And this is something that is characterized by the way you sample the tissue. And that is fundamentally different from the fact that this cluster is away from the truth, whereas this cluster is on it. So we're going to be talking about a bias, which is a systematic deviation from the true value, and we're going to be talking about the precision of your estimation procedure, which is the clustering around the samples that you have, the variability in the samples that you have. And the reason that you have these are numerous, and Susan and I are going to talk about these. And I can just say in the beginning, generally, these biases, this sort of systematic deviation, is the result of some assumption that one has made during the estimation procedure that is not true. And what's unique about the methodology we're going to be talking about here is that the stereological techniques, or the new stereological techniques, could just as well be called designed techniques, in the sense that they're designed to eliminate any assumptions about the size, shape, orientation, or distribution of the features that you're looking at. These previously had been the source of many of these systematic deviations, is false assumptions about the organization of the tissue.
So the design-based stereologic techniques are assumption free?
Yes, and that's why often they're called either designed based or assumption-free techniques, because they don't require any assumptions about the size, shape, orientation, or distribution of the features you're looking at. And that's unlike all of the previously available techniques, including the well-cited Abercrombie method. And we'll talk about that a little bit later.
So let's talk about where the variability actually comes from.
Yes, well, of course, there's in any sort of a type of an experiment, you're going to have an experimental deviation, and of course, that's we're wanting to know whether that's going to be important. And then, of course, if you have a number of animals in a study, each one of them represents a sample, and you could say that there's a cluster. There's a certain amount of variability inside of the group here. But there may also well be a bias in this, and it's difficult to know unless you've done things to assure that there aren't any biases in the way you're doing this, that this whole group may be shifted up or it may be shifted down by some sort of a bias. And in addition to that, we cannot assume that any bias that results in a systematic shift in this group is also going to be the same as the bias you'll see in this group, because the structure is essentially different. So we're going to be talking about the variability of a particular type of estimation technique, and we're going to be talking about why that might be shifted away from the true value. So in any study we have biological variability, and then we're going to have, in addition to that, this sort of variability that you see that is result from sampling. And you say, why is there variability in the sampling? Well, it's much like a public opinion poll. You don't ask everybody when you're trying to determine how many Democrats there are in the United States. You have a sample, and they ask that sample, and there usually is a margin of error about how accurate that is. So this is essentially what we would talk about, is the margin of error of this particular estimation technique that we're using here. And that's fundamentally different from a bias which might result in this cluster being up here or even down here.
So let's look at how bias is systematically different.
Yeah, so here's an illustration to try to show that as you sample tissue and you keep taking the running mean of the estimates that you make along there, if there is some sort of an assumption that you've made about the structure you're looking at that's false, you'll find that with continued or with extended sampling, then it's going to deviate from the true value, and it's going to deviate in a way that you will never really know how much this bias is unless you know what the true value is. And of course, that's a difficult thing when you're starting out. You don't know what the true value is. So you want to do everything.
You'll never know, right? You'll never know what the true value is unless you count everything.
Yeah, well, you can get a better idea. You can make a very good estimate by using unbiased techniques to make the estimate. Of course, the absolute value of it you will never know until you count everything. But you can get very close. You can get so that the margin of error of your estimate here is very, very small, such that you wouldn't consider it to be a major element in the comparison of two groups.
Right, right. So these biases are impossible to see.
That's the real problem with it. Unless you go back and do it with unbiased techniques and try to eliminate the biases, you'll never really know how true it was. So you can be in a situation where you can be making a biased and an unbiased estimate, and you may think just because the clustering is very good that you have a good sample here, and you may think, well, I have an imprecise system here and another imprecise sample, relatively speaking imprecise. But the point is that the unbiased estimates on average are going to give you the true value. You've eliminated all of the assumptions that are going to drive this in a biased way, so that you can actually change the intensity of the spots here by doing more sampling. But the point is, regardless of how much sampling you do, on average, this particular unbiased method is always going to give you, on average, the true result. This will not be the case with the biased system. You have, again, you can have precise and imprecise bias, but the reality is that when you're doing this for the very first time, you don't know where the truth is. And the only thing you can do here then is try to get at the places where there might be something that will create a bias in your data. And that's what we're going to really be talking about here for the rest of the talk. What sort of things enter into that? What sort of things you have to make sure you're doing in an unbiased fashion in order to be able to believe that you're actually honing in on the true value.
So to summarize, we want to ensure that we're using an unbiased methodology and employing it properly to make sure that we're up here in this top row. And then we'll decide on how much sampling is necessary to see how how tightly clustered our result needs to be towards the true value.
That's right. Ultimately, we can decide whether this sort of internal variability here in the methodology, if we know it's unbiased, we can then determine whether we should be here or here when we're designing our study, whether we want a lot of precision or not so much precision. but we definitely want to be up on the top line. So this illustration here is just really to make this point clear again. We're talking about the variance or the variability of an estimation procedure. Here you can see that we have an estimation procedure in which we have some red sections and some blue sections, and you can see that we can count the front ends of the objects that are inside of this region of interest to determine how many there are in the red sample and how many there are in the blue sample. And there are 66 of them in all, I can tell you that. And we look at how many of them have leading edges in the blue sample. There are 35, and in the red sample there are 31. So if you picked out one of these at random, you could say it's an unbiased estimate in the sense that on average it gives you the true value. The average 70/62 is going to be 66. So we could continue this sort of logic once more to just make the point about the fact that both of these are unbiased, although neither one of them gives you the true value. It has a certain amount margin of error, you could say, or variability or variance. The estimation procedure has a certain variance. So if we look at the next picture of this, you can see that in this case now there are 8 different samples. So we count the leading edges in the red and the leading edges in the blue and so on. And then we find out that the number of leading edges we see in each of these 8 samples varies considerably. And if we had picked one of these out by random and had multiplied it by the reciprocal of the chance of picking that out, that is to say we have 8 samples, so the reciprocal of 1/8 would be 8, this estimate for sample 5 would be 40. And we can see here there's a tremendous amount of variability here now, much more than before. We had very close values. They're only off by 4. Here we have a high value, 104, much larger than 66. We also have a low one here at 32. But the point I'm trying to make here is that this estimation procedure that I've used here, if I pick out one of these samples at random, I can consider that to be an unbiased estimate. But depending on how many samples I have inside this, it also has a lot of variability in the potential results that can come out from this. All of these are unbiased estimates because on average they give the true value. So on the next slide that I prepared for you, just to summarize this, because this is not the main point of the seminar today, but it's to make sure that people understand that you can have an unbiased estimate based on varying numbers of sections. And when you use very few sections, you can have a lot of potentially different types of estimates. But the point is that on average, any one of these sampling schemes is going to give you on average the true value. That is to say, if you'd used all the 96 sections that are in there. So the idea is that you can have varying degrees of variability in an unbiased estimation procedure. And it depends on how much sampling you do. So I want to make sure that one understands the distinction between this. These don't necessarily have to come very close to the true value at any one time. Here they're varying considerably, but on average they give the true value, and that is the definition of an unbiased estimate.
So we can estimate the precision of your estimate by using a formula called the coefficient of error, and I have this represented here as a circle around our actual stereologic estimate. So if you have a low CE, it might be a tiny boundary around your stereologic estimate. If you have a higher CE representing less precision, it's possible that you're going to have a greater circle diameter here. And we're only doing one population typically when you're doing a stereologic study per subject. And we don't know the true value. So you're going to end up with a single result. But you don't have this dartboard to show you where the true value of the true population value actually is. And so since we're only doing one estimate, even if that sampling is done with precision, so you have a nice low CE, it doesn't necessarily mean that that result is close to that true population value. If you have bias that introduces inaccuracy, we might actually be looking at this type of situation, which is going to cause us to draw some pretty poor conclusions. And so, as a reminder, the CE does not indicate that you are bias free. Let's pause for attendees to submit some questions if they have any. Karine has a question, Mark. She writes, this is a very naive question. To what extent can the sampling method lead to bias? In other words, what do you mean by a low CE? Well, I think those are two separate things.
Yeah, I think so too. The CE, of course, characterizes the reproducibility of a single estimate. It's just like with that colored egg there, you had a lot of different possibilities, and you could look at the distributions of the possibilities, and those sort of characterize the precision of the individual estimate. But then you have this issue of a group of samples or a group of animals or something like that, and the variability that's in there, because that variability is in addition to having any sort of stereological variability that's introduced by the estimation procedure, also includes the biological variability. So they're really two separate things. So the CE characterizes the reproducibility of the estimation procedure.
And I think that feeds in nicely to Pat's question. Pat asks if the CE is either the standard deviation or the standard error. And the answer is neither. The standard deviation and standard error relate to the group variance, whereas the CE is an attempt to estimate the variance of a single sample. Is that correct, Mark?
Yes.
Get that right?
Yeah, generally, the way you would calculate the coefficient of error, it's the square of the standard error of the mean divided by the mean. So it's put in relative terms. So it is the standard error of the mean. The standard error of the mean of repeated estimates. That is to say, you made an estimate inside of a rat hippocampus, and then you glued the whole thing back together again, and then you cut it up, and you stained it, and you did it once more, and then you counted how many were. And you keep doing this. You glue it back together again, you cut it up, and you look at how much variability is there in that particular way that you're looking at the hippocampus. And that's essentially what the coefficient of error is. And it's expressed generally in terms relative to the mean. So you get the relative size of this error. Much like you see with a margin of error, they usually put it in terms of a few percentage around the mean. So it's partially correct, but it's a variance that it's essentially expressed by the coefficient of error.
And it's predictive because you're basing all of that on a single pass through a region of interest.
Yeah, essentially.
Because of course you're not going to glue the hippocampus back together again and try again.
No, but that's what it represents.
Right.
It's a mathematical trick that allows you to see what would happen if you did that. And it's actually a very good mathematical trick. And if you want to see a reference to it, you can go to some of the papers that I've written with Lutz Lomianka, where we've talked about this issue.
And that actually hits very nicely with Philip Chu's question, asking if there are any published cases of unbiased stereology compared with total raw counts of a population. I think your paper with Lutz is 95 Neuroscience, right?
Yeah, I just don't have the reference at the top of my head.
That's okay.
But I can find
it on our website.
I can find it for you. I've got my book right in front of me here. But go ahead. That's essentially true, what you're saying.
And Rich Smynie has also published a paper where he's compared 3D serial section reconstruction to unbiased stereology. So if you're looking to see that stereology actually returns, actually works, those are both excellent resources for you. You can always contact us at MBF Bioscience, and we can point you to those articles. They're on our website as well, and also cited in various literature as well. So.
Thanks.
What do you say? Should we move on to our next question?
Okay.
Actually
we'll
the
go
next
to the next
topic.
part of the presentation. Yeah, I think so. We should move on.
So,
We can come back.
okay.
Yeah.
I said the next question because Yin Young Choi asks, how does one identify bias? Are there different biases? And that's exactly what we're going to cover in this next topic. So, what does cause bias in stereologic estimates? There are a number of ways that can lead to bias in your experiment, not just a stereologic experiment, but it relates to how you sample. So how you choose your animals. Obviously, you're very familiar with that concept that you need to choose animals that are representative of the population you're looking to learn something about. But now that you're doing stereology, you also need to consider how you sample those sections. So how you choose the sections, how you sample within those sections, both across the area and then through their depth. And then choosing the proper stereologic probe is critically important. Choosing it and implementing it correctly. And then so that falls under that broad category of how you're sampling. And then there's the category of how you quantify. And this leads to object and region identification. And so in this case, this is a prerequisite of not just stereologic experiments, but again, like choosing animals, it's necessary for any experiment that you're going to be able to do. You need to be able to unequivocally identify both where you're looking and what you're looking for, as well as the requirements that are necessary for the probe that you've chosen. So section orientation might be an example of that. Lastly, if you are using a method to quantify, you need to make sure that your system is calibrated and the requirements for use are understood and employed properly. So, Mark, let's talk about this.
Okay, well, here's a good example of the sort of biases that can creep in when you have to make assumptions about the objects. Let's say that you have a section that comes out of some sort of an organ, and you say, I'm going to use this very simple method. I'm just going to count how many profiles I see inside of this. This is potentially in danger of being a biased way of doing it, in that if you cut another animal or even the same animal or part of the other same animal in a different plane, then this particular way of quantifying the number of objects there are inside of all of this is not a very good one, because it's sensitive to the orientation of the objects. It only works if you have the same orientation in each section, and in generally not a good assumption in biological tissues. And as we talk about, and I know a lot of you are familiar with the optical dissector or the dissector way of counting, you know that it avoids this, because one section essentially samples in accordance with the size of the object, but it's also sensitive to the orientation. So the sort of techniques we're talking about, they avoid having to make any assumptions about this. Another type of assumption that is poor and going to lead to biases is that there has been, let's say you take a section out of a piece of tissue from an experimental animal, and it has 1, 2, 3, 4, 5 profiles inside there, or maybe you only see 2 of them because these are hidden when you look through it. And then you have your control animals, in which you have in reality the same number of objects, but they're smaller, and you count 2 profiles here, so that you conclude, oh, there are 3x objects in this animal and 2x objects in this animal. And that essentially, the difference here is a result of the bias in the fact that you're assuming that these are the same size. They're not. You're assuming that these are the same orientation. They're not. Or you're assuming that these are the same density, which they might not be.
And that actually can happen between experimental conditions, like your control and your treatment situation that you're talking about, or across developmental stages, but it can also occur within the same structure. Like in the example that you showed previously, where you had large cells, I'm going to say rostrally, but to the right or to the left, and then further back there were smaller cells. So even in the same region of interest, you can have a variance in the size or the orientation of the object that you're looking for.
Yes, that's exactly right, and that's one of the real big problems with using the older techniques, which are so-called model-based systems. They model all the objects have to have more or less the same appearance, and you could say, well, isn't it okay that I have the same bias in my control and my experimentals? And they say, no, actually, the bias might be very different because within the between the groups there may be changes that show that counting the number of profiles in a section is actually the wrong way to do it. It's going to lead to a bias.
And this also can show up with automated methods, actually, because with automated detection methods, you're going to provide a very strict set of criteria that you're going to use to identify your objects, and depending on that criteria, you may end up not being permissive enough to allow all the variation that occurs for the object that you're trying to quantify. That happens naturally.
You can even see from this example here, if you were to be using the dissector probe, which would allow you to avoid having to make any assumptions about size, shape, orientation, or distribution, you could say how many objects you're going to get per volume here. They're going to be 2. How many would be here? Well, you see one top here and one top here, and opposed to down here you have just 1.
Right, and is it?
And 2
it leads to some very complicated machinations to try to make sure that you're trying to remove some of these biases when in fact you can employ a stereologic method and remove these assumptions and make it easier for you to do your quantification and trust the answer that you obtain.
Exactly. Very good.
So what other types of biases that you need to be aware of when you're doing stereology? This is a very familiar bias, I'm sure, to a lot of people, this experimenter bias. And it's a conscious or an unconscious bias towards a result expected by the experimenter. And so this is true of all types of experiments, but and so therefore it is true of stereologic experiments too. So you may have an expectation that you or your PI wants to see a result in a particular way, that unconsciously you may support that in your data collection, not even being aware of it. And so ways to minimize this is to randomize your samples and to count blind. And when you're doing stereology, we're also going to suggest that you count at a minimum 100 probe and object interactions. And so that way you're ensuring that at a minimum you're encountering enough objects to be able to make a statement about what's going on in that particular sample.
I think the important thing here is making sure the study is done blind. I think that's the only real way you can get rid of this, is to have somebody doing this who doesn't know what the experiment is.
And actually that happens a lot at MBF Labs. A lot of the experiments that we run here at MBF Labs, I know nothing about the samples that I'm given. And so I have no idea what experiment was done. I just know that I need to quantify the number of neurons in the substantia nigra that are Th positive and those that are Th negative. And so each animal, we have no vested interest in what the answer is. We just need to create a unbiased and precise answer. Another bias that it can exist is an observational bias. And this is something that is familiar for those one representative section type studies. And this is typified by the streetlight effect. And so the streetlight effect is an observational bias where people only look for whatever it is they're searching for wherever it is the easiest. So the common story that comes along with this is a policeman comes across a person searching for something under the streetlight and asks, what's been lost? How can they help? He said he's lost his keys, and they both spend quite a while underneath the searchlight looking for it. After a little while, the policeman says, okay, well, is this where you lost them? And then why are you looking for them there? And he says, well, because that's where it's easier to look. And so this, the corollary to this in a biological experiment is to look in the area where you are very confident that you can delineate your region of interest. And so it's very easy to see the boundaries of the hippocampus at a particular level. And so you're going to choose to look only at that location. And so to minimize the observational bias from creeping into your experiment, you want to make sure that you first obtain consensus with researchers in your field, and that can be your colleagues, it can be your PI, it can be people that you respect, it can be object or papers reported in the literature on what your cell or blood vessel or whatever it is you're quantifying looks like in all of its many forms. So you got to get familiar with what it looks like with the staining that you're going to do. And then you need to understand how to delineate your region of interest. Again, you need to know how to find where you need to look, and you need to know how you need to be able to recognize when you found what you're looking for. And then finally, you need to ensure complete histological staining in these thick sections. If you can't see it, you can't count it, and that's an observational bias.
So the bottom line of this would be that all places of potential interest have to have an equal probability of being looked at, not only the places where the light is or where the stain is good or where there is no artifact or a fold. So this actually places considerable constraints on the preparation of histological material, because you have to be able to go into it and not only look at the bright places, but to be able to make sure that you have a sample that represents the entire structure. This is another example of type of bias that can creep into it. We call it methodological bias. You could see that if the thermometer has slipped down and is not properly calibrated, then you can be consistently making a systematic evaluation that deviates from the true value. So even though the individual measurements that I may make would vary by a little bit, this is just maybe the parallax of where I have my eye when I'm looking at this. But it's very different from the mean value you're getting in this, and when you're looking down at 80 degrees. And these sort of biases can creep in from the improper calibration of your equipment. The lenses and the magnification that you're using, the ability to focus through a known thickness of section on the z-axis, and all of these things, if they're not properly calibrated, they can result in systematic deviations from the true value. Regardless of how much you really want to do this well, it's always going to be away from the true value. So you have to check the stuff out from the start. And this is really something that you can go back once if you find out there's an error. For example, somebody else might publish something with a different value, and then you might go back to find that in reality you could have corrected for the lens and the oil that you're using, then make a small difference in that. So you can actually go back and take the data that you had before and make corrections for it. But you have to also be aware of the fact that those biases exist if you're going to be able to do that. So as pointed out here, calibrate your system, obviously, and then practice measuring the things like section thickness and so on, to make sure that you're sort of in concurrence with your colleagues about reading. Where you're actually reading here is what is the value here.
Yeah, just like you learn to pipette by pipetting a known amount onto a balance to make sure that you're actually using the Eppendorf pipetter correctly. It's the same sort of thing when you're going to be using your microscope to quantify your histological specimen. Okay, so now we're getting into the meat of the matter, which is sampling bias. Sampling bias is a bias that's caused by choosing a non-random data for statistical analysis. And so, as you might imagine, this is the crux of the situation when it comes to stereologic experiments. And what we're going to encourage you to do and show you how to do is to develop your sampling scheme properly to ensure randomization at each level. And this can potentially exist at all levels of your sampling scheme. It can start with the choice of animals, how you sample those slides and sections, how you sample with your stereologic probes, and even the orientation of the plane of section. And so um, this bias exists due to the flaw in the sample selection process, where a subset of your data is systematically excluded due to a particular attribute, and that can produce distorted results. So we're going to spend the majority of the time today talking about sampling bias. So how do you obtain a representative sample?
Yeah, well, that's a good question. The bottom line for all of this, for a representative sample of whatever feature you're looking at inside of this, is that you have to have a sample of sections that potentially, or let me put it another way, all potential sampling sites within the region of interest have to have an equal probability of being in the sections that you're going to use. So we're talking now about sampling sections. And you don't always want to take the first section. You want it to have a random start and then a certain interval after that. And that's to ensure that you have a sampling scheme that on the bottom line will ensure that all parts of the structure have an equal probability of being in that set of sections. That is not to say that all potential regions inside of the entire structure are going to be on those sections, but everything inside there has an equal probability of being in this sample of sections. So you do that either by pulling these out randomly or systematically randomly, which I'm sure many of you are familiar with, how to sample sections in a systematic random way. So if you don't do that, if for example you always start at the very beginning and you have a structure that has a big gradient again it, you're actually going to bias sample because you're always sampling in the very first section. And that means that there are certain sections, section 2, 3, and 4 perhaps, that never get sampled when you're using this particular method for selecting the sections that you want to use. So the idea is you want to have a system that allows you to pick out a set of sections in which all regions of all parts of the region of interest have an equal probability of being in those sections. And then if you can't, if the section is too big or contains too much information, you have to do some sort of subsampling of that when you're trying to decide where are you going to put your probes inside of this. And this also has to be either a completely random choice of positions within there, or it has to be a systematic random. That is to say, another way to ensure that once you have the sections, that all parts of the section have an equal probability of being sampled by your probe. So that means that when you're down at this level, you're actually still making samples in which all parts of the structure potentially can be present in that section. So that's how you do that. You have to have random sampling inside of the series of sections and within the section itself. And we're not talking about the probes themselves, we were talking about the position of the probes and where they're going to be. So that has to be done. And in addition to that, the number of probes that you put in to any one section has to be proportional to the area of the section. So you don't always want to have 5 probes on each section. The number of probes should be proportional to the area of the region of interest. And that generally is done automatically for you with the automated systems and so on. But you can do it by simply randomly placing a transparent grid on top of a section and then just marking out the places where you're going to do that. Again, use the same grid on each section so that the amount of sampling here is proportional. If it's not, you're going to have a biased sample. If you're always looking at 5 fields in each one of these sections, maybe the structure itself is only half as big up at this level. So this is
so you're sampling more of it at that.
So it's not representative because the bottom line is here that you want to make sure that all parts of the structure have an equal probability of being sampled. And if you're sampling more on one of the sections, then you're breaking that rule.
That also is appropriate when it comes to thick section probes where you're dealing with a height sampling fraction. And in this case, what's typically done is you measure the section thickness, and then in the case of the optical fractionator as an example, you're going to size your dissector plus the guard zones to fit within that section thickness that you obtain. What can happen though is that you can encounter a section thickness that is less than what you set your parameters to. And that causes the guard zone and the dissectors to no longer fully fit within your measured section thickness. And what this means now is that you're no longer sampling this location with the same parameters or probability as this previous site. And so in order to avoid this, you need to have a good understanding of how much variance you have in your section thickness, and so that you choose your height sampling parameters appropriately, and that's the guard zone and the dissector height. And so one way that you can do that is to perform a comprehensive pilot study and measure at each counting site or at many counting sites so that you get a good understanding of how variable your section thickness is. You need to make adjustments early and recount when necessary because this is a bias that can't be corrected. And another way that you can account for this, and this is something that we do at MBF Labs for our pilot study, is initially we count without guard zones. So we just mark all of the objects that meet the criteria to be counted through the section thickness, and then determine what the appropriate guard zone and dissector height is for each animal and apply those values post hoc. And so in a sense, we're determining what the proper height sampling fraction should be after we've collected the data. We've gotten a lot of questions since we last paused, so let's pause and address some of these questions. Here's a question for you, Mark. Doesn't the optical dissector assume homogeneity of cells? If the cells are clumpy, you may get a wildly incorrect number. How should one address this? Should I delineate an area focused around the labeled cells, i.e. remove unlabeled space that may inflate cell count estimates? And the answer is no.
Yeah, my understanding is that the question is that does the distribution of the objects affect the unbiasedness of the estimator? Is that a fair way to reformulate that? Because the answer to that, of course, is no. I don't want you to go all the way back to that colored egg illustration, but if you try to recall that in your head, there were structures with different shapes, orientations, but also with different distributions. In one end of the egg there were many small objects, and the other end there were very big objects. So the answer is that the only thing that will perhaps be affected is the variance of your estimator, but not the unbiasedness of it. And I can go on to explain that more if you would like, because if there's a lot of clustering and you have a lot of blank areas, you could say that you might have a lot of variability from one slide or from one individual to the next. But it's going to be only the variance of the estimate you make that's going to be different, not the unbiasedness of it. So it could be if you've got a lot of...
you're affecting the precision.
The precision of a non-homogeneous structure, if it's being sampled in a homogeneous way, is going to have a higher variance than a structure that has a homogeneous distribution. But both are going to be unbiased. One will have more variance than the other because you have a lot of blank fields, perhaps.
Right. All right. Here's, I think, perhaps a related question. And this person asks, I would like to know how to count cells without bias in a transplant in the striatum. We saw that in the cell transplant area, the cells are much too close and there is difficulty to count.
Much too close. Why does that create difficulty? It's possible to count very highly compact regions. For example, the granule cell layer of a mouse hippocampus or the granule cell layer of a mouse cerebellum. I doubt if the proximity of the cells in this transplant are going to be closer than those. So what you have to do is develop the right stain in order to be able to see them. Now you're going to count nuclei, and there has to be some space around the nucleus that's not going to be stained if you stain for the nucleus. So what I would suggest is you have to go back to the preliminary pilot stage of this and try to figure out what stain allows you to resolve those neurons, and there has to be some stain that allows you to do that.
And it may be a combination of stains.
Yes.
So you may need a nuclear stain in order to allow you to disambiguate closely spaced objects, but you may need a marker that allows you to see which cells were transplanted. So maybe they're GFB transfected or something like that. And so you can combine labels and make your counting criteria a nucleus that is surrounded by your reporter label.
Yes, that's the way to do it. One way you can sort of get more definition if you're using this sort of classic initial types of stains is you can use some sort of RNase to remove the RNA staining that's in the cytoplasm and then just have the nucleus remaining. And that's what we've been able to do in some of our preparations, is that to be able to get the resolution to distinguish between cells that lie on top of each other. Can be done.
Yep, and also I would add in a system requirement and make sure that you're counting at a high enough magnification so that you are able to have a very thin depth of field.
That's right.
If you are counting, if this material is in bright field, you might use an oil condenser, for example, to help increase that depth of field, or decrease the depth of field a little bit. If you're doing fluorescent material, you may consider a structured illumination system or a confocal system, which really will allow you to have very crisp and clear visualization of your structure and objects of interest. If you are using a confocal, though, you have to make sure that it has the ability to collect that systematic and random sample that we talked about in the previous slide. So you need to be able to randomize where you're looking. And so you can use software like Stereo Investigator that will control the confocal microscope to ensure that it goes to those systematic and random locations. But it is not sufficient to just acquire the stacks on your own and then count them with a dissector. There's been a couple of questions about how many people should count a specimen. Should it be, is it important to have 2 people count blind? Or put another way is, would using the central limit theorem by utilizing multiple people doing stereology on the same sections eliminate any of these biases?
Well, yes, of course. That would be helpful, but it's probably not a very efficient way of doing it. I think the best thing to do is get standardized before you start and then have one person do it. Everybody has a little bit different way of looking at things, and I think that it's much better to have one person do the entire study rather than have a lot of people, one person do one animal and one person do another animal. I would never recommend that. But if you want to have 5 people do the counts in order to get correspondence, if you have the resources for that, I would say fine, do that. But generally that's not the case, and I think what would be appropriate would be for people to spend some time, those who are interested in the issue, the authors of the potential paper or something like that, sit down ahead of time and agree about what's going to be done and just assume then that that's going to be done. Of course, there could be a bias in that. We don't know. But then again, one can say that perhaps it's better, if there is going to be any bias, that it's the same bias in each one. It would just add a lot more noise to it to have 4 or 5 people do it. But they should essentially end up with the same result, and they should agree upon that. should be able to agree upon that.
Right, right. And that's where coming to consensus is important. So that falls under that observational bias and training. So if all the folks in your lab are come to an agreement about what you're doing, then that is one way to ensure that you are making strides towards minimizing or eliminating that bias. We have a lot of questions, but we have a whole nother section to get through. So I'm going to press ahead and we'll see how things pan out as we get to the end. So now let's talk about utilizing probes to ensure that you're looking at your tissue in a representative way.
Okay. Well, generally the recommendation is that when you're estimating volumes, you use point counting, and when you're estimating surfaces, you use lines, and when you're estimating lengths, you use surfaces, and when you're estimating number, you use volumes. Now, different biases can creep into these different types of probes. And just for an example here, the idea is now to take to count directly how many objects there are in a volume, and using the dissector method. Now, a bias, and we've already talked about this, a bias might be introduced here if you didn't take into account the size of the objects. You just said, I see 1, 2, 3, 4 objects inside of this, and maybe some of them actually are split between this section and the next section, so you're going to be getting a bias in there. And we talked about this sort of a bias before, one counting profiles as you look through it, the other one counting a unique point, sort of like the leading edge as you focused down through this. Now, for potential biases that can be entered in with these are that there is a requirement for the use of linear probes and surface probes that they have to have a isotropic interaction with the structural feature that they're interested in. And you could say if the structure was itself isotropic, it wouldn't make any difference how you place this linear probe in or this surface probe within it. But we know that the vast majority of biological structures, if not all of them, are not totally isotropic. So when we're talking about improper use of a probe, bias can come in if we don't take into account the fact that these linear and surface probes have to be oriented randomly in the side of the tissue in order for the stereological formula to work. So that this places an additional constraint. So you say, oh, well, I'm going to assume that the capillaries in the neocortex are isotropically oriented. That is to say, capillaries go in all directions within the neocortex with equal probability. And let's say that it's not true, and let's say that you just say it doesn't make any difference then, since the tissue is isotropic, what way I section it. And you would end up with a bias if in fact it is not isotropically oriented, which it most likely is not isotropically oriented. You don't want to have to assume anything about the orientation of the structures. So there are a number of ways that you can do this. Either the structure can be assumed to be isotropic, which is probably a poor assumption and may lead to some biases.
So let's not do that.
So we want to do that. Or you can do something with the way you cut the tissue, or you could do something with the type of probe you're using so that it would ensure this isotropic interaction with the feature you're looking at.
Right. So just to have a couple of mental images about what we're talking about here. To achieve sampling isotropy, like Mark said, the object orientation is one consideration. And so you don't want to make any assumptions. And so it's safest here to assume that the objects have a preferential orientation. This is especially true with length, especially in the brain. This looks remarkably like any anterior commissure or any other white matter tract that you're used to looking at. You know that the fibers don't orient themselves in a random way in those particular locations. But it would be true of cell populations as well. And so unless you want to prove that your object is itself isotropic, you're going to need to achieve that isotropy in another way. And so that can be how you prepare the tissue. And so you can create isotropic tissue sections, or you can create vertical tissue sections, or you may prefer to use preferentially cut tissue sections. So, for example, coronal. And you may want to use this because you need this type of orientation to be confident in how you're going to delineate your region of interest. If this is your situation, you need to make sure that you choose a probe that allows you to achieve isotropy, because preferential cut sections obviously are not random. And so you have then to achieve your isotropy by using by relying on the stereologic probe. And there are a number of isotropic probes which free you from having to prove that either your object is isotropic or to make your tissue isotropic. Examples are the optical fractionator, and Spaceballs are two examples. We gave Dan Peruzzi and I gave a webinar devoted to how to choose your probe, and so you can review that that webinar if you'd like to go into this topic in more detail.
Yeah, well, this slide sort of going back a little bit about something we talked before, and I was talking about a dissector probe. I'm assuming that the vast majority of the people listening know what a dissector is, but I'll just say very briefly, you have a physical dissector, which is two separate sections and defines a volume. Essentially what you try to figure out is which objects belong inside of the red section. That's the sample section. So if it was in the middle of a structure like this, you'd have the red one and the blue one right next to it, and what you do is you try to look for the leading edge of an object, that is to say an object that's on the red one but not on the blue one. So this is the physical dissector, or the dissector principle, and what it does is it eliminates any necessity for knowing anything about the size, shape, or orientation. Here's what I was referring to earlier, this green egg with the small structures down here, some along this way and some are along this way, and then you have the same up here with bigger ones. And you can see when you use exhaustively the dissector technique through this, it doesn't make any difference about the size, shape, or orientation of it. Each structure is reduced to a leading edge which is non-dimensional. So essentially what you're counting are the leading edges here. And I think most people could see, I don't know why you'd have to do an experiment to prove to yourself that the dissector is working. I know that this question was asked before, but I think when you understand the principle behind it, and if you fulfilled the only requirement that there really is for the dissector, that you have to, all objects have to be potentially identifiable. You can't have any of these that are not stained and try to make some sort of conclusion about that. But this is then the physical dissector, and of course it is we'll talk in a second about the optical dissector, which is simply nothing more than a bunch of optical sections that go down through here. But the point I'm trying to make is here, if you made a serial reconstruction going through this and found all the profiles that ended up being connected with each other, you'd end up with 66 objects here. Now, if you were using one of these other ones and you say to yourself, okay, I know because I've done this that there are on average 5 profiles per object. If I take the total number of profiles and divide it by 5, I end up with the right number. So it can work, but I have to know how many profiles there are per object. And if I'm wrong about that, you can see very easily I can get a bias here. And how do I really know this in the population I'm looking at? Here I could actually go in and measure each one of them. 66 objects is sort of like something that you're able to do. But if you're talking about structures like in the human hippocampus with 16 million structures, then you don't want to have to go in and figure this out and open yourself up to the possibility that there is a bias in it. And in addition to that, you can see, how do you measure this in your tissue? How do you measure this in the real tissue? And it depends on the orientation of the structure. So you're out here having to assume that, oh, now I'm going to see how many sections there are when I cut it this way through a normal nucleus, and then the other way, and it's going to be different. So the dissector method gets rid of assumptions that can lead you into major biases in your results. And this historically, I think, has been one of the real problems with quantitative neuroanatomy, is that biased techniques have been used. People have made assumptions about the size, shape, and orientation in order for these applications here to be used, and they vary a lot. Nobody can agree about them to such a degree that many people don't even want to read papers that have to do with quantitative morphology. So we're in the process now of trying to repair that by showing you that there are ways that you can count that are not biased by the size, shape, orientation, or distribution of structures.
Mm-hmm. And that's also true when it comes to section plane as well. It doesn't matter how you count how you cut the region of interest if you're doing it properly and getting all of the region of interest having an equal opportunity of being sampled. It doesn't matter how you cut it. If you are counting the leading edge, or the first time that object appears as you encounter it, you'll obtain the same answer.
Yeah, that's right. So you could say this is really one of the best examples of a so-called design-based stereological technique. The probe itself is designed in such a way that it requires no assumptions about the size, shape, orientation, and distribution of it. So we're really talking about assumption-free methods, or reducing the assumptions by designing the probes and the sampling scheme in a particular way. And this sort of logic continues on down to if you have a section in which there are all too many objects to count, you can do a subsample within this, but you also have to do that subsample of the ones you're going to see. Is it here and not up here? You have to select the ones out you're going to check to see whether they're here and not here. So you have to subsample this area. And the way to subsample that is with a counting frame that many of you have probably seen in the literature, but don't fully understand why it's like this. And this is the same counting frame that is essentially virtually moved down through the tissue to make an optical dissector. And then you have the counting rule. If it's hitting the yellow, if the top of the object is hitting the red line here, then it's not going to be counted. And you can say this is an unbiased counting frame. Hans-Jørgen Gunnarsson published this many years ago. That why is that? Now you can see what would happen if you covered this entire section with unbiased counting frames that were completely adjacent to each other, or as they would say, you tessellate the entire structure. And what you'll find that this is sort of a proof for why this is an unbiased counting frame, because when you count everything with this unbiased counting frame, nothing gets counted twice, regardless
and
of
everything gets counted once.
And everything gets counted once, regardless of its size, shape, orientation. So here we are now getting down to the level of sampling in the section itself, and we want to do a subsample, so we have to have an unbiased selection of the objects that we're going to sample and test to determine whether they're going to be counted or not. So I think that's it's necessary. If you don't use that counting frame, I think there are theoretically other counting frames, but if you just use a simple box and say anything that touches the edge is going to be counted, or if only the things that are entirely with inside are going to be counted, then you're going to end up with a biased count, because that's not an unbiased sample of the profiles in the section. Yeah, so I think we've made this point before. If you're going to do a proper unbiased stereological estimation, you have to have access to the whole region. And I underline the word access. You're not going to necessarily look at all the sections, but you have to potentially from the start have the ability to pull out a set of sections that is such that all parts of the structure that you're interested in are in those sections with equal probability. And we went through that once before. Generally, you would say that you can make an unbiased estimate on the basis of one section. It's generally not done in biology because
There's too much variability from
Yes, you could
location to
imagine that in
location.
that colored egg that I've been using, there are 96 different sections there. You pull one of them out at random and you measure the number of cells in that, and then you multiply it by 96. You're going to get a tremendous amount of variability. Some are going to have really a large number and a really small number. So that's to say that you can have an unbiased estimate from one section, but generally speaking, in biological structures, you have to have a certain number of sections, and we're not really going to talk about today how many you need, because there's no set answer to that. There is maybe some rules of thumb, but this will have to return on another seminar. And random sampling is the important thing here, but systematic is even better. And if you want to read about it in my book, you can see why systematic random sampling is better than independent random sampling. And as I pointed out, you have to have an isotropic interaction between the probe, the object of interest. This is a particular importance when you're using linear probes and surface probes.
Right, right.
And of course,
And then, of course, you need to have a unique point to recognize your object of interest when you're counting. And I'm just going to add one here that's not written, which is you need to make sure that your histology is properly prepared. So to avoid those observational biases, you need to make sure you have complete staining through your entire section thickness, for example. So to avoid bias in your estimate, you've got to be able to identify that object and region of interest. You need to make sure that you've got unbiased sampling at all levels of your sampling scheme. And you need to make sure that your measurement instruments are functioning properly. So make sure that they're calibrated. If it's a stereo investigator system and you need help with that, just contact us. We're very happy to do so. And how do you do these things? So you seek expert advice, and you get input on your design to make sure that you're approaching things in a way that is an unbiased way. So randomizing your samples, counting blind, all the way through to determining how much sampling is necessary to return an estimate that's going to be relevant to the question you're asking. You need to provide good, thorough training, not just for you, but for anybody else who's going to have a hand in your experiment. So if you're going to have a student, a grad student, or even your PI count your tissue for you, you need to make sure that they are stereologists so that they know what they're doing. And finally, do those quality checks on your data as they come in. So don't wait till the end to analyze your data and find out that your section thicknesses are wildly variable or are twice as thick as what you cut the section thickness at. In the first case, that's a sampling bias because your dissector height is going to be inappropriately sized, most likely. And then in the latter case, it could be a methodological bias because you may have the step size of your stage set wrong, or you may have your objective lens defined wrong, so you're returning an inaccurate thickness. If you want to learn more, you can go to Mark's website, neurostereology.info, or communicate with him on his Twitter feed. You can attend a course taught by Mark at Neuroscience this year in DC, or you can come to the Marine Biological Laboratory in Woods Hole for a more intensive class. That's a full week, right, Mark?
Yeah, 5 days with a lobster dinner.
Oh, I know. I remember that lobster dinner. It was delicious. And I didn't come just for the lobster, I swear. You can get great stereology information and material on stereology.info. We've done a number of webinars, and we're always looking to find out what you are interested in hearing about or learning about next. So when you leave the webinar today, you'll have an opportunity to provide us with some comments and suggestions for what we should do next and what you would like to see from us in the future. We do list references on our website as well as within our software, Stereo Investigator. So if you're using Stereo Investigator, you can access some publications the citations right within our software. But you can also see references that have used stereology right on our website. It's been a wonderful discussion, I think. I've certainly enjoyed myself tremendously.
Thank you, Sue. I also enjoyed it. I think it was good. And I'm glad to have this opportunity to talk about this topic. There's unfortunately still too much bad stereology being done. I feel really bad when I sit reviewing articles and have to tell people that there are problems with the way they've been doing this that raise questions about the validity of their data. It's not something I like to do, but it does happen. But is more and more good stereology happening, but still a lot of bad stereology. So I hope that this forum that Michael Brightfield is presenting here can sort of continue to reach out to the community in a different way than normally through these courses or through publications. So thanks very much.
Yeah, I think all sources are great ways to sort of build your knowledge and make you a more well-rounded scientist. So I think you can gain a lot from courses and a lot from reading on your own, but sometimes it's best just to do it yourself and see what you get.
Yeah, and even if you're not going to ever do any stereology, I think it's important that you understand when good stereology is being introduced in a paper, so that are they really using unbiased sampling schemes and so on all the way down through it. So I think even though you're never going to perhaps use this yourself, you can now evaluate a lot of the literature with regard to sort of quantitative morphology in an appropriate way.
Maybe what we should do is after this webinar, maybe I should get your input on how to read a stereologic paper to know if they're doing good
Yeah, well, I'd be
stereology.
happy. That would be fun to do that.
Yeah, okay. Well, we'll do that, and then I'll write it up, and we can put it on our website so other people can hear
Yeah,
your words of wisdom.
great. Love to.
Okay, so the questions that we didn't get to, we'll make our way through them. There are a ton, so it's going to take a little while for them to get on our website, but we will work on it. I really appreciate everyone's attention and diligence and staying till the end. I really appreciate it, especially since we were a little beyond the time that we had initially specified. So fill out that survey if you wouldn't mind. I'd like to know what you want to hear about next. And until next time, you can reach us at www.mbfbioscience.com. You can contact us. You can send us an email at info@mbfbioscience, or you can give us a call. And we'd love to hear from you. Take care.
Bye bye.